Assalamu alaikum students. Before going to our educational topic, please subscribe my channel Muntzir Mehdi Science TV on YouTube. Press the like button and share this lecture to your friends and classmates who have no access to it to add in their science education. And those students that are preparing privately can benefit from our channel very effectively. Let's come back to our topic, that is inhibitors. Students, these inhibitors attach with the enzyme, so in reality these are known by the name of enzyme inhibitors. It is an important, very important question for annual and supply exams. Firstly, we see its definition and then I will talk about its types. As its definition is representing with the help of its word inhibit, which means stop, check, prohibit, and suppress. So there are many words which can define the inhibit mean. So inhibitor means a substance that stops and checks an activity, a function of a substance. Enzyme. Here that substance means enzyme. So enzyme will that particular substance that will act with an inhibitor and will influence by the effect of inhibitor. So we call it as enzyme inhibitor because enzyme is used in this inhibition process. So students, this topic is also known by the name of enzyme inhibition. Dear students, what is the role of the enzymes? To catalyze a chemical reaction, to speed up a chemical reaction. But the function of the enzyme can be stopped, retarded by changing pH, proper temperature range, and various other types of chemicals. So if we change the required optimum pH of an enzyme, required optimum temperature of an enzyme, then the function of the enzyme can be stopped, inhibited. On the other hand, if we use certain types of chemicals like cyanide, that is a poison, then the activity of the enzyme can also be inhibited. So students, there are two ways to stop the activity of the enzyme. First one is by changing the temperature or pH, and the second is by using a chemical agent. So students, by the action of a chemical, an enzyme can also stop its function performance. So in this topic of enzyme inhibitor, changed pH or temperature will not act, but a chemical substance is utilized to study the enzyme inhibitor. So here is the function of a chemical substance. So when a chemical substance reacts with the enzyme in the place of a substrate, and that particular substance will not change into product, but it only blocks the enzyme's active site, is an inhibitor. Dear students, this is the main definition of the enzyme inhibitor, during which an inhibitor attaches with the enzyme's active site in the place of substrate. Then this inhibitor will not transformed into product because this inhibitor comes only to block the enzyme's active site to stop its function. Students, enzyme inhibitor can block the active site temporarily and momentarily or completely and permanently. Its examples include antibodies, antimetabolites, poisons like cyanide and snake poisons, whose example is venom. In addition to all these examples, some drugs can also function as enzyme inhibitors. So these are the names of some chemical agents which can stop the enzyme's activity. This is the whole definition of the enzyme inhibitor. Next, we see the definition of enzyme inhibition. Students, enzyme inhibition. Inhibition is that process during which the activity of the enzyme is stopped with the help of a chemical substance. So that whole process during which the activity of the enzyme is stopped by the use of a chemical agent in the place of a substrate at the enzyme's active site, and that chemical agent will not transform into product, is known as enzyme inhibition. Students, next we talk about the types of enzyme inhibitor. First one is irreversible enzyme inhibitor and the second type is reversible enzyme inhibitor. To understand irreversible enzyme inhibitor, let's suppose it is our enzyme and it is our irreversible inhibitor. This irreversible inhibitor occupy the enzyme's active site and will remain in this attaching form, or it may destroy the active site of the enzyme permanently. So students, during this attachment of the enzyme and inhibitor, strong covalent bond is formed. So covalent bond that is not easily breakable is present in between the enzyme and the inhibitor chemical. So due to its presence, students, which presence? The answer is that the presence of the covalent bond. This type is irreversible inhibition, during which an irreversible inhibitor attaches with the enzyme with the help of a covalent bond, and that inhibitor that attaches with the enzyme with the help of covalent bond is known as irreversible inhibitor, because neither covalent bond breaks easily nor enzyme will able to catalyze. Students, if you want to watch the covalent bond more clearly, then you can watch it here at this site. So suppose when an inhibitor attaches in the active site like this, then this type of covalent bond is formed in between the enzyme and inhibitor. As in the active site, the covalent bond does not clearly visible. So after the inhibitor attachment, the enzyme takes this form and will destroy permanently and will become virtual to attach any substrate molecule. So students, we can say that the shape of the enzyme has changed to this form and its organized configuration is denatured, and this denatured enzyme can't re-adopt its previous configuration. So students, now active site is permanently occupied with the inhibitor, and eventually due to its presence any substrate can't able to attach with it, because active site has already occupied with the inhibitor. So if a substrate is come, then where will it attach? As we can see that active site is not available, so this inhibitor will call an irreversible inhibitor, and the process will call as irreversible inhibition. As enzyme can't re-adopt its denature configuration to the organized configuration, so this process is irreversible inhibition. As this inhibition happens only in one direction but is not reversible, so it is called as irreversible inhibition. Next, we see the reversible inhibitor. That what does happen with the reversible inhibitor? To understand it, suppose it is our enzyme and it is our inhibitor that attaches with the enzyme in the place of active site. But students, the bond between the enzyme and inhibitor will weak. So these signs show that the bond between the enzyme and the inhibitor is weak. It may be weak hydrogen bond or hydrophobic interactions, but it can't strong covalent bond. Mostly we call it as hydrogen bond that can be easily and automatically breakable. As covalent bond cannot automatically break, but it is the possibility of the hydrogen bond that easily break. So the inhibitor, after the breakage of that weak hydrogen bond, can leave the enzyme's active site, and then this active site is free to attach any substrate. So reversible inhibitor attaches with the active site only for short time period, and after its release the enzyme becomes free for a substrate and again starts its function. Students, why is this inhibitor called as reversible? The answer is that, as we have seen, that due to the presence of a weak bond, often inhibitor separates from the enzyme and the enzyme starts its function again. So this inhibitor is known by the name of reversible inhibitor. Dear students, keep this important point in your minds that this reversible inhibition process can also be done with the help of increasing concentration of substrate. which means that when the concentration of the substrate is increased near a reversible inhibited enzyme, then this increased concentration of the substrate causes the inhibitor to release from the enzyme, and then the enzyme is become free for its function. Students, this point often happens as MCQs during exams, that by which method a reversible inhibited enzyme can be restarted, or what is the method to separate a reversible inhibitor from the enzyme. Its answer is that by increasing the substrate concentration. Next, reversible inhibitor also has two types. First one is competitive inhibitor and the second is non-competitive inhibitor. As compete means competition contest, because from the word compete the word competitive has derived. Students, competitive inhibitors normally have structural similarity with the substrate. For example, this is our enzyme, this is our inhibitor, and here the substrate is in this form, that both inhibitor and substrate are similar. Both have triangular shape, so they are not alike but only similar. Both have resemblance with each other, as they are not 100% alike but only resemble. So, due to its resemblance, its shape, similarity, both start a competition, compete with each other, because substrate try to bind the enzyme's active site. At the same time, inhibitor also tries to bind the same active site. This binding, this attachment depends upon the amount of substrate or inhibitor. If substrates are more in amount comparatively inhibitor amount, then a substrate will attach to the active site. But if inhibitors are in more amount as compared to the substrates, then an inhibitor will attach to the active site. So it creates a competition in between the two. The substrate with deficient amount will not occupy the active site, whereas the inhibitor with the deficient amount will also not occupy the active site. Only the sufficient amount substance, substrate or inhibitor will win the competition and will succeed in occupy the active site. So when inhibitor will bind the active site due to its more amount, then it will occupy the enzyme's active site. So there will not remain any chance for the substrate to bind here, as it is not empty. So due to the presence of the inhibitor, the enzymes will become functionless. So here a question happens, that can the inhibitor catalyze, as it has attached to the active site, so it definitely it must catalyze. But students know this inhibitor does not catalyze, it only inhibits the function of the enzyme, because an inhibitor is selectively selected by the binding site of the enzyme, but they are not able to activate the catalytic site. So when catalytic site is not activated, then product is not formed. So we can say that by the attachment of the inhibitor, it will not split, but it just stops the enzyme's catalytic activity by taking the place of the substrate. In this way, the function of the enzyme is inhibited. Dear students, this whole mechanism is also known by the name of competitive inhibition, and we can reverse this process by increasing concentration of the substrates. So when we provide abundant amount of substrates near the enzyme, then the inhibitor will release from the enzyme and chance becomes for the substrate to attach the active site. Students, next we study about the non-competitive inhibitor. Dear students, non-competitive inhibitor, as its function is representing by its name, that it does not show any competition between the substrate and inhibitor, because this substrate does not have structural similarity with the inhibitor in this case. For example, this is our enzyme and it is its active site. Here substrate attached. Dear students, instead of active site, an enzyme also possesses another site whereof name is allosteric site that is present here at this point. We can define allosteric site as the site other than active site on the enzyme upon which certain effectors can bind is known as allosteric site. So students, allosteric site is a specific site on the surface of some enzymes for the binding of effectors or inhibitors. As the binding of effector or inhibitor causes a change in the enzyme structure, as this is a substrate and this is an inhibitor. So in this case, the inhibitor shape is totally different from the substrate. In this case, the inhibitor will not bind at the active site but will attach at the allosteric site. Due to this attachment, students' active site receives a signal. So due to its signal, the active site changes its shape in this way that now it is unable to bind any substrate. Now this substrate can't bind with the active site. So now the active site will remain totally free. Students, next we see some differences between the two. The first and main difference between the competitive and non-competitive inhibitor is that competitive inhibitor attaches at the active site, whereas non-competitive inhibitor attaches at the allosteric site. Second difference is that in the case of competitive inhibition, the active site is not free. The inhibitor is attached. On the other hand, in the case of non-competitive inhibitor, active site is free, but although the enzyme has changed its shape due to the attachment of inhibitor at the allosteric site, so substrate can't bind at the active site. So now the poor substrate is lonely and friendless. As substrate is deprived of an enzyme, so it is friendless. That is the second difference between the two. The third difference between the competitive and non-competitive inhibitor is that in the case of competitive inhibition, the enzyme does not contain any allosteric site, whereas in the case of non-competitive inhibition, the enzyme has allosteric site. So students, these are the three major differences between the competitive and non-competitive inhibitors. Students, this difference is often asked during the exams, that what is the difference between the competitive and non-competitive inhibitors and what is the difference between the irreversible and reversible inhibitors. So now you have efficient knowledge to write the answers of these questions in your own words. Students, now we again take a summary of this topic, that the enzyme inhibitor is a chemical substance which stops the catalytic activity of the enzyme by attaching with the enzyme's active site in the place of substrate. So temporarily or permanently the function of the enzyme is stopped. Students, next we have seen two types of enzymes inhibitors, that were irreversible and reversible inhibitors. During irreversible inhibitor, that once stopped the enzyme's activity, then this activity can't be reversible. That's why it is known by the name of irreversible inhibitor, as the bond between the enzyme and inhibitor was strong covalent bond. But in the case of reversible inhibitor, the bond was made weak between the enzyme and inhibitor, so this weak bond can be broken down easily, so there will be chances for the enzyme to start its function again. Next, reversible inhibitor is of again two types. First was competitive and the second was non-competitive inhibitor. The competitive inhibitor are resembled with the substrate, so it was attached at the active site. But this inhibition can be reversed by increasing the substrate concentration. On the other hand, non-competitive inhibitor was attached at the other allosteric site, so the enzyme's shape was changed. Therefore, the substrate can't bind the enzyme. So, students, it is a very important topic that often comes during annual exams as a large question or as a short question. That what is the enzyme inhibitors, or differentiate between the irreversible and reversible inhibitors, or differentiate between the competitive and non-competitive inhibitors. Students, one of all these three questions can come as a short question. Prepare it ably so that you can write it at your paper. Dear students, as I have told you some main differences between the competitive and non-competitive inhibitors, here is now your test that write some main differences between the irreversible and reversible inhibitors. That is your test. Now write the answer of this short question. Students, at the end I want to encourage you that take care of all of your things like books, notes in your file, dresses, and shoes. Place them at their proper place organizingly and orderly so that you avoid by the harm of ransacking of your things. Lastly, many thanks for watching this your video.